Hello everyone. So today we are going to learn about negative staining. So negative staining is a type of differential staining method which we use to visualize the structural differences between the organisms. Okay. So in the differential staining we have learned about separation into groups. One type of method was separation into groups and the other was structural visualization. Okay. So this thing we have studied in our previous video, in the staining video. If you have not watched it, the link is in the description below. So this is the negative staining. It is a structural visualization based technique. So what are the uses of this negative staining? If you see, it is used to observe the intact microbial structures without disturbing its cellular morphology. So the cellular morphology is not disturbed here. Everything remains intact. It allows the visualization of shape and size of the organism against a dark background. Now, there are bacteria that are very difficult to stain, right? So, they are not stained using the gram positive, gram negative and the acid fast. So, the negative staining, what it does, it shows the structure, the shape, the sizes. It allows the visualization of the shape, sizes and the arrangements of the bacteria that are very difficult to stain. Example is spirulina. So here in this picture we can see that this bacteria are not stained and they are appearing as a transparent or translucent organisms against this dark background. So it provides a detailed information of the features like flagella, fimbriae, pili, their arrangements and their orientation. So in gram positive, gram staining and acid fast staining, what we see is that it divides the bacteria into different groups, either positive, negative, acid fast or non-acid fast. Okay, we have seen. It does not allow the visualization of the shapes or structures like this, like flagella, like fimbriae, pili. Okay, all these structures are not visible, but in the negative stain, it allows the differentiation of these minute structures also. Okay, so that is why negative staining is very important. It is one of the easiest and rapid staining method used in microbiology. Besides this, it is also used to visualize dilute suspension like food particles, liquid crystals, liposomes and cellular fragments. Now the scientists who have developed this are Sidney Brenner and the other one is Robert W. Horn. So both the scientists have developed this staining method in the University of Cambridge in England in the year 1959. Okay. Now the scientist Sidney Brenner, he is also a Nobel laureate. He has shared a Nobel Prize in Physiology and Medicine with H. Robert Horwitz and Sir John E. Sulston. Okay. So now this staining method, why it is actually called negative staining? Okay. So because the bacterial cells are not stained by the dyes, only the background containing the cells are stained. That is why it is called negative staining. Here in this picture, we can see there is a negative staining of the cryptococcal capsules. So this is the coccus in shape. We have studied about it. All these bacteria have not containing the capsules have not taken up the stain. Only the background is taken up the stain. So that is why it is called negative staining. In this picture, below this picture, we can see there is the bacillus capsules, which is showing the same translucent pictures against the dark background. Okay. So because of this non-staining of the bacteria or the capsule, it is called the negative staining. So this negative staining, it requires the use of acid stain like the India ink or the nigrosin. Okay. So this acid stain, there are three types of stain we have studied: acid stain, basic stain and the neutral stain in our staining video. So you can watch it. The link is in the description. Since it is acid stain, it has negatively charged chromogen. So that is why it cannot penetrate the bacterial cells. Why? Because the bacterial cells also have negative charge surface area. So that is why It is called negative staining procedure. Okay, so the stains that are generally used for the negative staining are India ink, nigrosin. So nigrosin is the most commonly used stain in the negative staining. 10% of nigrosin is used in this staining. Okay, so the other stain that are used are phosphotungstic acid, uranyl acetate, ammonium molybdate, etc. So this ammonium molybdate is very rarely used because of its charging issues. Otherwise, phosphotungstic acid and uranyl acetate are also commonly used besides nigrosin. So coming to the negative staining procedure. So in step 1 we take the slide and we grease free it using alcohol just to avoid the distortion of the smear. Okay, so this grease free this step is very important to avoid the distortion of the smear. Means the smear should not get distorted when we make it. Okay, so that's why this step is very important. In step 2 what we do is we put this drop of nigrosin or India ink on one edge of this clean slide. Now after that what we do, we add a loop full of bacterial culture into this stain drop. Then we mix this stain drop with the bacterial culture using the sterile inoculating loop. We'll move it in a rotary motion and we'll mix it well. Then after that we'll take up another clean slide and put it over the stain, making a 45-degree angle. Now after this we drag this second slide over the first one to its edges, making a thin film. And after that this thin film is allowed to air dry and then it is observed under bright field microscope under oil immersion. Okay, so oil immersion uses 100x. So in 100x we'll see the structure. Now this procedure will become more clear when you see the pictorial representation. So before this we need to see the market availability. This is India ink and this is nitrosin which is available in the market. Now coming to the representation, here we take the grease free slide and then what we do, we put a drop of nigrosin or India ink at one edge of the slide just like this, and after that we put a loop full of bacterial culture over this drop and we mix it in a rotary motion using a sterile inoculating loop and we mix it very well. And after that we take up another clean slide and put it over the first one at a 45-degree angle. And what we do, we just drag the second slide on the first one in somewhat in this manner as shown in this picture, and then we make a thin film. And after this the thin film is allowed to air dry and after that it is observed under microscope. So using oil immersion. Okay, so this is the whole procedure of the negative staining. Now what we will observe under the microscope? We will observe that the capsule bacteria, what will they do? They will not retain any stain and hence they will appear translucent against a dark background as shown in this picture. Whereas the non-capsule bacteria, what will they do? They will retain the stain and they will appear black in color like shown in this picture. Here in this picture we can see the bacteria that contain capsules, they are showing some halo zone around the bacteria, along the bacterial cell, whereas the non-capsule bacteria, what they do, they are stained, they take up the nigrosin stain and the structure cannot be differentiated with the background, right? So this is how this will appear. Now in this staining method, one step is avoided, that is heat fixation. So this negative staining do not apply heat fixation in order to avoid the shrinkage of the cell. Okay, so this heat fixation step which we have seen in our separation methods, we will not use in this structural visualization method. Okay, now coming to the mechanism of the negative staining. So the mechanism depends on the charge of the cell wall. Earlier, all the mechanism basically depends on the composition of the cell wall. In this negative staining, it depends on the charge of the cell wall. So the use of the acidic stain, what it does, it creates repulsion between the negative charges of the stain and the bacterial surface, rendering the dye to not penetrate the cells. Here in this picture we can see this is the bacterial surface is negatively charged, and the acidic stain, that is nigrosin, which is black in color, is the acid stain. It is also negatively charged. So the bacterial stain negative, this acidic stain negative, what they do, like charges repel each other. They repel each other, which we have studied in our physics class also. So the like charges repel each other. So that's why the bacterial cell does not take up the stain of the nigrosin, of the negative stain, right? So the background takes up the stain, but the bacteria will not take. So in this way we can distinguish the structure. We can visualize the capsule. So as a result, we'll see a clear cell with a dark background. Now coming to the capsule staining method. So the capsule staining also employs a negative staining. So it's not different from negative staining, only a small part is extra in this capsule staining. So before moving forward, we need to understand what is a capsule. So the capsule is the gelatinous extracellular layer surrounding the bacterial cell wall. Okay. So generally it is made up of polysaccharide, but some are composed of polypeptides. The capsule stain, they are very poorly stained with general staining reagents. So therefore it implies acidic stain or basic stain or both. So this negative stain which we use in this capsule staining as well, it allows the visualization of unstained capsule against the stained background, right? So since these capsules are very fragile in nature, they are destroyed by heating. Therefore, the heat fixation step is avoided in this method. Now coming to the reagents which are used in this staining method is crystal valor 1% and nigrosin 10%. So the procedure of the capsule staining, as I have mentioned earlier, it is similar to that of the negative stain. Only a small part is added. So we'll quickly review it. So we first put a drop of nigrosin on a grease-free slide. Then we add a loop full of bacterial culture onto the stain drop using inoculating loop, and we mix it properly in a rotary motion to form a smear. Not a large one but a small one. We mix it properly. Then what we do? We put another slide over it, making an angle of 45 degree, and we drag it on the first slide, over the first slide, making a thin film. Okay. Now this thin film is allowed to air dry for 5 to 7 minutes. Now in this step, we make sure that we do not heat fix. It's a very important step. We have to avoid the heat fixing step, right? So after that, what we do after the drying, we add the crystal violet onto the smear. We cover it fully and we allow it to stand for 1 minute. And after that, we tilt the slide at 45-degree angle just to drain out all the crystal violet from the slide. Then after that, it is air dried and observed under the microscope at 100x. So this crystal violet step is the extra one which is added in this capsule staining. So the observation under the microscope, what we'll see is that the bacteria will take up the crystal violet, whereas the background is stained with the nigrosin and crystal violet, mixture of nigrosin and crystal violet, like shown in this picture. And here the capsule will not take up the stain, and therefore it appear like halo. So this capsule will appear like halo zone against the dark background, while the bacteria take up the crystal violet. Now here we need to remember that not all bacteria have capsules. So some of the microorganisms that have capsules are Streptococcus pneumoniae, Haemophilus influenza, Klebsiella pneumoniae, Pseudomonas aeruginosa, Cryptococcus neoformans, and Neisseria meningitidis. Okay, so these are the bacteria that have capsules, and the bacteria that have capsules are pathogenic in nature. So that's why the differentiation of the bacteria based on their external structure, that is the capsule, is very important, and this could be achieved by the negative staining method. So in this video, we have learned about negative staining, its procedure, its mechanism, and capsule staining.